The transport across the cell membrane consists on the movement of ions or molecules inside or outside the cell through its plasma membrane. In this transport, the channels of the cell are open depending on its needs. They sometimes use energy, ATP, as we know it, to make this transport through the membrane happen. A hypotonic solution is when there is a lower concentration of the solute, which are ions and hydrophilic molecules. Hydrophilic molecules are molecules that are attracted to water. In this case, water comes to it. A hypertonic solution is when there is a greater concentration of the solute and water moves out of it. It is hydrophobic because it doesn't attract water. So, in conclusion, the water moves out.
Animal cells have specific characteristics in the cell membrane. They are composed of phospholipids. The inner part of the cell membrane is hydrophobic and it is nonpolar because it has cholesterol molecules, which are lipids, which means fats, like oil, therefore it repels the water. The polar region of the cell membrane is hydrophilic, polar, which means that it attracts the water. Each layer is called a leaflet. Gases like oxygen, water, some proteins, amino acids, and also some lipids are molecules that can pass freely across the cell membrane. The materials we were going to need were the two eggs which we were going to experiment with, the acetic acid, which is basically vinegar, corn syrup, which was the hypertonic solution, water, the hypotonic solution, clear glasses where we could see the eggs, a weighing scale to weigh our eggs before and after the experiment, phone to record the videos and pictures, and our computers so we could gather all our data. Egg number 1, which was the one that was going to be added in the water, weighed 100 grams originally, and egg number 2, which was going to be added in the corn syrup, weighed 75 grams.
2 eggs?
Give it all away. Catching fire as the wind blows. I'm a better original.
After 3 days, Fabricio took out the eggs from the vinegar and rinsed them with water, and now he's adding them to both our solutions with the corn syrup and the water. After spending 8 hours in their solutions, egg 1 was actually weighing more, 10 grams more, which meant that it absorbed some of the water, and egg number 2 was shrinking, which meant that it released some of the water and it weighed about half of its original weight. Now, in this part, the eggs are being switched from places, so now the egg that was in the corn syrup is going into the water, and the one that was in the water, it's going into the corn syrup. After waiting another 8 hours, we saw that the egg that was previously in the hypotonic solution but then was switched to the hypertonic solution actually lose some water, which meant that its water molecules were going out, and vice versa. The one that was firstly in the hypertonic solution and then was switched to the hypotonic solution gained some of its weight back. With this experiment, we concluded that everything that was happening that led the molecules come in and out was due to osmosis. The cell membrane had a channel that led molecules either in or out, and in this case the hypertonic solution was making things come out of it, in this case water molecules, and the hypotonic solution was letting things come in. By firstly putting the eggs in the acetic acid, which was the vinegar, we got rid of its calcium and we were able to prove the hypothesis we wanted to, and it was correct.